Good. Excellent. Okay. So, hi, everyone. Thanks for attending this month's webinar. We have, I will be leading it today because our normal leaders are at the Festival of Genomics and talking about our Common Fund data ecosystem resource. We'll be talking today about some new methods that we've been developing that will hopefully show up in the portal soon. So I think I'll get started. For those who don't know me, my name is Jason Flannick. I am the technical and scientific lead of our knowledge portals, and I also have a lab at Boston Children's Hospital where we do a lot of research on statistical genetics, and it kind of intersects with our interest in the knowledge portals and developing methods for integrating various types of data to calculate genetic support, which is also a major component of our portals. So I will be talking today about some new methods that we've been developing that should be appearing on the portal soon, and you'll also get access to kind of a private link that's not yet available that you can play around with and give us feedback on. Okay, so what is genetic support, first off? You probably all have heard this term. It's most commonly used in drug target validation. And I think the distinguishing factor about it is that it's really gene first. So most genetic studies are phenotype first. You start with people with and without a disease or people with a phenotype, and then you're looking for variants and genes that can explain the phenotype, and you kind of take that wherever it leads you. Reverse genetics or gene first approach begins with the gene that you discover or are interested in for other reasons. And then the goal of genetic support is to see whether human genetic data supports a role for that gene in disease. And we're very interested in this question because we think that this is the most common use case of our portals. There's many people who have genes that they're studying, and determining whether there's relevance to some human phenotype we think can add a lot of value as those people are making decisions about whether to pursue those genes further for whatever reason. So a couple years ago, we published an essay that outlined an argument for using genetic support. And it was based on a couple of very simple rules that we termed the huge calculator. This lookup table summarizes it. You basically get a probability that the gene is involved in disease that is based on the strength of rare coding variants in the gene, and also based on how close that gene is to a genome-wide significant GWAS SNP. And there's a couple of categories based on how close it is to that SNP. But basically you get this table, and this is implemented on the portal. It's the first thing that you see when you go to the gene page. And I think I'm showing, I actually don't remember what gene I'm showing the huge scores for here. Oh, CCND1. And one of the first things you can see here on the bottom is that we break apart the support into a common variant Bayes factor, a rare variant Bayes factor, which we have these categories for how strong those are, and those map to probabilities in a way I won't get into today. And on the top, and then you have this combined score that combines both of them. And one of the first things that you'll see on this plot is that it's very sparse and it's very discrete. And if you've used the portal, you're probably very familiar with this. And I'm going to talk a little bit today about why this is and how we're addressing improving that and some methods that we have underway. And I'm going to call that improvements to this is a direct evidence model. So I'm using this term direct evidence because the huge calculator says, is the gene supported? Is there support for a role for this gene in a trait, given the associations I observe for it? I'm then going to spend most of the time today, though, talking about a new indirect evidence model, and I'll describe what that means. And it's something we're very excited about. And I think we'll have a lot more interesting statistics and hopefully be just interesting from a scientific perspective in addition to a resource on the portal. So first, though, I just want to talk a little bit about 4 improvements that we have made and are piloting to this huge calculator, which currently measures direct GWAS evidence. I'm just going to go through the intuition for this. So there's actually 4 areas that lead to that kind of sparsity and discreteness. So one issue with the current calculations is that the support from a GWAS association is mainly due to whether that gene is within some fixed window of a GWAS hit. And so I'm showing here kind of like a box that indicates how this works. Essentially, every gene within this box on the bottom of this genome-wide significant SNP would get the same huge score of 3. And then if you are one base pair outside of it, you'll get a huge score of 0. And so obviously this is, a little bit severe. So we've relaxed this constraint to have a more continuous notion of distance from a GWAS SNP. And I won't go into the details, but essentially there's this nice continuous decay function that we fit such that on average, the closest gene has about a 70% probability of being, having support for a given SNP. So that's one more continuous extension we've made. A second extension is to account for the number of genes that you have at a locus. So again, right now, the huge calculator kind of analyzes each gene independently. But if you kind of look at this top locus compared to the bottom locus, you would probably intuitively think that, these genes at the top have probably less support for them than this gene PROX1 on the bottom, because PROX1 is kind of on an island. There's not many other candidate genes there, or at least there's fewer. And then on the top, there's more candidates who are kind of competing for the signal. So we've made some changes there that now we jointly calculate values for all of the genes within a locus as opposed to each gene one by one. A third change that we've made is rather than simply looking at distance to the lead SNP, we look at distance to all of the SNPs in the window. So you can essentially think of this as analyzing not just the lead SNP, but the credible set of variants at the locus, and kind of marginalizing or integrating over all the credible sets SNPs to kind of allow for loci like this, where the signal is actually spread across a fairly large region, and therefore there's more support for a gene like MYL7 or GCK than you would think based solely on proximity to this purple lead SNP here too. And then the final most significant change is relaxing this so we're able to calculate support for genes that are not within genome-wide significant loci. So this is the insulin receptor signal for diabetes in 2018. This is not in a genome-wide significant locus, so it would have a huge score of basically 0, and there'd be no support given to this gene. But obviously, you're seeing here there's a near genome-wide significant peak. And so there's probably at least non-zero support. And so we've come up with a statistical generalization to account for this as well. So I just wanted to briefly kind of go over that, because all of the kind of work I'll be showing the remainder of this webinar is going to use those revised, more continuous notions of direct genetic support. These changes are not a full statistical method. We actually have a method under development that we'd be happy to discuss and share. There's a postdoc, Alex Yamas, in the lab who is working on that. But they maintain most of the advantages of the existing huge score that if you've used it, hopefully you like, which is that it's very fast and interpretable and easy to use. So that's kind of it on the direct genetic support extensions. And I want to spend most of the time today talking about indirect evidence. And I'm going to define what that is. And by the way, please, if you have any questions along the way, just pop them into the chat. We should have time at the end for questions, but I will definitely kind of be checking the chat and taking questions along the way. Okay, so in terms of indirect support, what do I mean by that? So returning to this insulin receptor signal, and also I'm going to show another example here, which is the HNF1 alpha region. Again, these are both from the 2018 diabetes GWAS. And in case you're not familiar with type 2 diabetes as a trait, insulin receptor is the receptor for insulin, which is obviously the dominant molecule involved in type 2 diabetes. And the HNF1 alpha is a gene that has variants that are responsible for a very common monogenic form of diabetes. So these are both very well-known diabetes genes. And the point is that you probably, if you knew that and you looked at this peak I'm showing at the top, you're probably more confident that that's really a real signal that just hasn't reached genome-wide significance yet for various reasons. Than if this were just gene X, and on the bottom, even though there's this is a signal, and we know it's not always the closest gene, right? And there's 7 genes nearby. You'd probably wager a lot of money that HNF1 alpha is the causal gene at this locus, right? And the reason for that is that you have a lot of prior information. And prior information, I'm using this on both a very technical sense, which is information that you observe before genetics that gives you a prior probability that you think the gene's involved in disease. But also informally, just as like, in the ad hoc way that we all do when we look at and evaluate genes, we kind of Google and read the literature and read papers and have our favorite genes. And there are some we favor more than others. And, for those reasons, these two signals, really the support for these two genes are much higher than just the direct scores would indicate, right? And that's really what we're trying to get at when we talk about indirect genetic support and this method PIGEAN that I'm going to talk about today, the name of which will become clear when when I give you one interpretation. Essentially, PIGEAN is trying to solve this question of how we take all this heuristic and unstructured information about genes that are not related to genetics, and then integrate that information with the genetic data that we observe for a disease, for a gene, to get at an integrated estimate of genetic support. So the direct evidence comes from the GWAS hits and the exome hits that we get for the gene. The indirect evidence comes from everything else that we know about the gene. Okay, so the way that we model this unstructured or prior information is through gene annotations or gene sets. And so I'm going to use those terms interchangeably. So you can think of as annotation as any value you can calculate from a gene. So this could be like the expression pattern of that gene in a given given tissue. It could be like the results from some, differential expression experiment. But most commonly, it's done from gene sets that are curated in the literature or in a lot of databases that kind of give groups of genes that have a similar property. So one of the favorite ones that we use are mouse phenotypes. There's a great database that curates the literature. And for all these 11,000 phenotypes from the mammalian phenotype ontology, they record all of the genes that have been published to have that phenotype in a knockout mouse model or any mouse model. So that would be an example of a gene set that's a gene annotation. The genes that have that been reported to have that mouse phenotype all have value 1 for that phenotype, for that annotation, and all the others have 0. So in practice, that's a very sparse matrix if you think of like for each gene, all of the annotations that it has. And then there's other databases that you're probably all familiar with, like MSigDB is this database that is commonly used in like differential expression, gene set enrichment, that is a compendium of all sorts of, databases like the Gene Ontology and Wiki Pathways and all things like that. There are text mining you can get from, co-occurrence of genes with various terms from PubMed abstracts. There's network neighborhoods, like all of the binding partners for a gene from databases like String. There's, gene expression patterns. So a tool that I'll talk about in a minute called POPS has aggregated a ton of single cell data and clustered that. So you have, which cell types given genes are expressed in. And then our colleague Avi Mayan from, who works with us on Common Fund ecosystem has a whole collection of hundreds of thousands of gene sets from this resource called Harmonizome. So the point is there's like hundreds of thousands of these annotations that have been curated, and you probably have all seen them before, and you've probably run gene set enrichment analyses and you get a ton of stuff. And some of it makes sense, some of it doesn't. You do your FDR, and that's a very common thing that's being done. But the issue that we have with incorporating these to prioritize genes is that it's very hard to know from these hundreds of thousands of genes what matter for a given trait. So again, I'm using type 2 diabetes as kind of a running example. So apologies to those who are not familiar with that trait. But this is an example of a gene, FOXO1, that was in our essay that I referred to at the start of today, and just kind of what the raw data looks like for the mouse phenotypes that's observed for it. So each of these columns is like a different mouse annotation. And at a glance, some of them look relevant to diabetes, like, improved glucose tolerance. But others are less clear, like pericardial infusion. Does that matter? So we got to figure out which ones of these matter to not just diabetes, but whatever trait we're evaluating. We have to somehow quantitatively compare them. So it's not, maybe decreased energy expenditure and improved glucose tolerance both matter, but maybe one matters more than the other. And then more complicated yet is then what happens when you observe multiple? Like if I, observe both normal glucose homeostasis and normal gluconeogenesis, does that count twice, or is it kind of like the same phenotype that I'm measuring? So all of those are kind of the challenges in kind of converting this huge amount of data into support for a gene. And this method called POPs, I think, had the idea that we're going to use here today for doing this. And so basically the idea is that given that you have an annotation or a gene set, you can calculate whether that set is enriched for GWAS signals. And in principle, and specifically, they use a method called MAGMA that we have on the portal that calculates gene scores from a GWAS, and then it uses that to calculate enrichment. And then I'm showing here on the left, the p-values are kind of what these enrichments produce. And I'm just showing some positive controls, all these mouse gene sets involved in insulin and glucose, which are diabetes, obvious relevant traits. And you can see that the p-values are significant, which indicates that there's information in these enrichments to help prioritize these gene sets. And so then what POPs does is, based on those enrichments, it does this feature selection where it essentially tries to use the enrichment scores to predict whether, they try to fit a joint model that uses all of the features to predict the gene's MAGMA score. And from that, they get this ranking of genes in the genome that is basically kind of like the sum of all the enriched features that a gene has. And because they're fitting this model jointly, it's a way to kind of control for, the annotations that are dependent or independent from one another. And so basically this is how it works. You kind of use these MAGMA scores to guess at the genes. You then fit the joint model. It's regularized using an algorithm called ridge regression that, if you're not familiar with this, it kind of controls overfitting. And then critically, they use this cross-validation method where you fit the model for leaving a chromosome out and then make predictions for that chromosome. And POPs is really exclusively used that then within a GWAS locus, you then look at the gene with the highest POP score to prioritize it. And this is a very widely used method. I think it was cited almost 100 times before it was accepted. And it's appearing in a lot of the effector gene lists that we have on the portal. So that, I think, is encouraging and, indicates that I think people are seeing the value of such an approach to help prioritize genes at GWAS locus. But there are some limitations, especially as it pertains to our goal of enfolding this into a genetic support system on the portal. So first off, the top POPs genes don't match the curated genes lists that we have. So for type 2 diabetes, we have a list of about 133 genes that were curated by our collaborators, Mark McCarthy and Uba Mahajan, as well as a subset of those that are very, very likely to be causal. And you can see by this Venn diagram that a very small fraction of these are in the top 100 POPs genes genome-wide. One of the reasons for this is that POPs is very biased towards well-studied genes. So there's a very strong correlation to the number of annotations a gene has and how prioritized it is. And in part, this is because it's using MAGMA results, which are noisy. I'm showing here like a locus where the signal is very broadly spread. And what MAGMA does is gives a lot of genes in this locus very high scores, where in reality, probably only a small number of these are actually the true gene. And that's obviously going to degrade the quality of your enrichments. But really, the main issue for our uses is that it's not answering the question of genetic support. POPs is only used for producing rankings of genes within a locus, which is fine, but the majority of genes that we care about, especially in this gene-first approach, are not in GWAS loci. What we want to be able to do is, for any gene and any trait, be able to say the extent to which these annotations and its GWAS associations support it. And POPs is kind of, we can't really do that here because these scores can't be folded into a model like that very easily. So that's where PIGEAN comes in. And I'm going to kind of gloss over the statistical details, but I'll give some intuition as to what it's modeling and how it's working. So basically, this is, if you're familiar with the way that this graphic, each of these circles is a quantity that we're trying to calculate, and then the shaded in things are things that we observe. So we have values for every gene in the genome, and that's what we care about. This is, you can think of as a 1 or 0, whether the gene is involved in a trait. Okay, so it's a very simple model. The gene is or it isn't. Okay. Then we have circles for each annotation, and the annotations have values that correspond to the extent to which they increase the probability that the gene's involved in disease. So I think that's somewhat intuitive, right? Like you saying, I think this gene has a 5% chance of being involved in disease. Now I've observed this, it has in this annotation that increases or decreases my probability by some extent. And we don't know what those values are, but at least that form of the model, I think, should be pretty intuitive. And then we have the data, which is shown here on the top, this capital D, which are the associations we observe for the gene. And intuitively, you can think of that the more likely a gene is to be involved in disease, the stronger the associations we'll observe for it. Okay, so think about this as like a feed-forward model where we have some annotations. They increase or decrease the probability the gene is involved in disease. That then gives a probability for each gene, and then the gene probabilities further inform the genetic associations we observe. So that's why these arrows are going in this direction. You kind of specify the model in this like feed forward way, because that's easier to model. But then, and I think I have, okay, so, and then the way that model says is that we impose this distribution that most of the gene sets don't affect the probability at all, but some do. They add, as I said, and then the data that we observe for the gene, that's where our huge model comes in. That these edges, this distribution is specified implicitly by the huge scores that we have. But then what our goal is actually we want to do inference in the reverse order, right? So we specify the model going up in this diagram, but we observe the genetic associations and we want to calculate the gene and the gene set values given the observed genetic data. Okay, so if you don't follow that, that's okay. It's the intuition is just that we are trying to calculate these gene and gene set values jointly, given the data that we observe. And the way that works, this is, I think, a more intuitive way, is that you have all your genes, and then there are these gene sets that kind of group genes. And presumably, most of these gene sets are not random, but they're, grouping functionally coherent genes. And then you get a guess at which genes are relevant by the huge scores. And then you run your enrichment that finds these gene sets that are enriched. And this is very similar to what POPs does, But then you re-estimate the gene scores now that you know that the genes in this gene set are more likely to be relevant. Now, once you've done that, all of a sudden, this second gene set now might look interesting. And now I can re-estimate that, and again, re-estimate my genes. And you basically are iterating this back and forth where you're re-estimating your genes and your gene sets. And it might seem circular, but there's actually a very, very sound mathematical justification for this. If you're familiar with the approach called Gibbs sampling, that's what this is doing. Gibbs sampling samples from this joint distribution that I mentioned we want to calculate by doing iterative conditional sampling. And all of that are details I'm going to sweep under the rug, only to suggest that that's kind of the intuition as to how this is working. And so just to kind of give you some intuition as to what exactly this thing is doing. So you can actually view this as a statistical generalization of POPs. That's why I spent a fair amount of time on that. If you kind of don't do this iterative update process, but just kind of fix your observed gene scores for all of the genes not on the chromosome of the gene, and then just do a single shot Gibbs estimation, you actually get a method that's very, very analogous to POPs. There's another method, and this is where PIGEAN gets its name from, as principled priors for the gene. So if this calculation in the upper left, this quantity is what we care about. This is the probability that a gene is involved in disease, that's the DI, given the gene sets and the observed data. This factors into two terms. One is the observed data, which is what we have on the portal now, which is the huge scores. And if you recall, I mentioned the huge scores on the portal are what are called Bayes factors. And that's kind of like the degree to which you update your prior. This second term, which on the portal now, we just use a fixed prior of 5%. Actually, what PIGEAN is doing is calculating that prior, not just to be fixed at 5%, but being calculated conditional upon the annotations, where it's kind of learning that prior dynamically as well. So that's why this term priors inferred from gene annotations came from. And then as the most, I think, kind of coolest interpretation, you can think of this as the indirect half of genetic support. So again, what we have today is support for a gene given directly the associations observed for it. That's what this like gene 1, given just its observed associations. The other gene associations in the genome actually also can support that gene if the associated other genes share gene sets with that gene. So like through this network, this like indirect evidence, these genes indirectly support the gene you're looking at by kind of propagating back through this network, through the shared annotations and up to the gene. So ultimately that gives us, the direct evidence for the gene from the huge scores, then the indirect evidence from the gene sets. So just before I show you kind of this in action, just to give you some validation that it's working somewhat well. When you run this, as I mentioned, you get a parsing of the support for a gene into its direct evidence, which I'm calling GWAS here, the indirect, which is the prior, and then the combined, which is both together. And this is sorting for diabetes, the genes that have the strongest direct evidence. You can also sort by genes that have the strongest indirect evidence. And of course, you can sort by the genes that have the strongest combined evidence, which is kind of your default. And I'll spend much more time momentarily on exactly what these means and how to use them. Is there a question? Absolutely. Please ask if there's a basic. I'm sure other people have the same question. Okay. Well, please put it in the chat if you do. I will read that and I will answer it. Okay. So in addition to estimating these gene scores, you also get the gene set annotations. And so one key thing here is that these annotations are fit jointly. Okay, these are the effect of the gene set accounting for all other gene sets. And I'm kind of showing here a ranking of the top ones for diabetes. And these can be quite different from the top marginal effects. That's the effect of the gene set in isolation. And you can see a lot of these. And usually when you run a traditional gene set enrichment analysis, you get marginal estimates. And this one on the right would be much more familiar to people who do gene set analyses for diabetes. On the left, though, you see that the joint effects can be quite different. And just for those who are statistically inclined, this is entirely analogous to, on the right, the p-values you observe for a SNP, which is its marginal. And on the left, the joint is what you would do if you did a fine mapping and a credible set, once you try to like parse those out. Okay, I'm confused about the source of the prior information. So the prior information, the method is agnostic to that, but we have compiled these annotations as our prior information. So we curate from the MGI database 11,000 mouse phenotypes. And so for each of those phenotypes, you have the list of the genes that are in that phenotype. So conversely, for each gene, you have all of the mouse phenotypes that have been observed for it. So that would be the prior information from that mouse data set that you had for the gene. Then we also are showing all of these others below. So we also run text mining, for example, for each gene, get all of the terms that it occurs with. We have prior information from its expression patterns. So that's what I mean when I say prior information. It's all of these annotations or gene sets that the gene is in. And on the portal demo I'll show, you actually will have the ability to kind of use only some sources of these this information. Okay. How do the scores change for genes in specific cell types and tissues? So sorry, hold on, okay. So that is an interesting, so if by scores you mean annotations, the annotations obviously will change for like these annotations, single cell gene expression directly change based on the expression pattern of genes, right? So there is one data set that is annotations for the expression in islets, and obviously those will be highest for genes active in islets. and others in adipose tissue. For these other gene sets like hallmark adipogenesis, that's a pathway annotation that will obviously be higher for genes in the tissue in which that pathway is active. But a priori, that's just the information from the annotations you have. When we then are going to combine all of that information with the GWAS to kind of identify which of these annotations matter the most. And then obviously, though the degree to which those gene scores vary based on cell type will depend on the degree to which that cell type or that tissue is active in that gene. So it's a very complicated integration step that's combining these annotations that track with tissues with disease that tracks with tissues, and kind of how that all comes together. I'll allude to very briefly at the end, but it's something that we're actively trying to kind of parse out and tease out. Okay, will PIGEAN help to pick out the genes with GWAS signals below the genome? Yes, absolutely. And that's, in fact, one of the major motivations that we have had for doing this. So in a minute, I'm going to show. So actually, if you look at this insulin receptor signal, this is the 2018 type 2 diabetes GWAS. And this is kind of the hallmark for why we were interested in this sort of method. Like, obviously, this gene is relevant to disease. And if you actually, I don't think that gene itself appears in the top list here, but is insulin? Yes. So you can see here, insulin receptor here has very weak GWAS support. So it's a 38% probability, which is higher than the background 5%, which is kind of what's reflected by the fact that it's not genome-wide significant. But the prior from the annotations are so high that the combined score, it actually is like the 5th highest score in the genome for indirect support. And then when you go to the combined score, it's actually in like the top 15 genes. So if you look on this combined ranking, you'll see some genes that have absolutely no support, like the AdipoQ, that are not near a signal at all, but have such higher indirect evidence that they actually have a very high combined score. Now, obviously, the combined score is a simplification. There's a reason that some of these genes don't have GWAS signals, and you probably don't want to treat them equally, right? Because there is something to be said for a gene having direct support, but it's also the case that genes that have indirect support can still be relevant to the trait and may just not have a GWAS signal for various other regions. And that's exactly a major goal of what PIGEAN is doing. It's also, as I'll show, it's kind of picking out at a GWAS locus which genes have the highest prior, and therefore using that to kind of rank genes compared to each other. And those are the two major ways it's kind of integrating this information. Okay, good. I was wondering how your method can account for the heterogeneity of effect size estimation due to genetic ancestry. Your GWAS data may come from non-European. Yeah, so right now, that is not something that we are considering. So if you had heterogeneity at the annotation or pathway level, we are going to be estimating that averaged over all of the data that is input into the model, right? So if you're hypothesizing that, like, for South Asians, like the lipodystrophy annotations East Asians, lipodystrophy annotations are more important than the insulin ones, and you are feeding it like a multi-ancestry or European data set, you're going to find mechanisms and genes that are relevant to that trait. Now, one thing I would say, though, is that the huge score does not require linkage to disequilibrium. It's still based solely on proximity to a GWAS hit, which is a limitation, but actually a strength when it comes to handling transethnic data, because you don't need to worry about modeling LD. So it's kind of more robust to differences in ancestry in that way. But certainly, if there are different mechanisms, you would get different results in how you run it. And I think exploring that will be an interesting question. I don't know about others, but I probably have a hypothesis that mechanisms are more likely to be shared across ancestries than variants are. But that would be an empirical question that we would have to look into. Okay. So one nice thing about, so I mentioned, so we get the gene values, which are kind of two axes. We have the gene set values, and then you get this nice estimate of provenance. So you don't just get the indirect support for a gene like GLP-1R, you get the specific gene sets that supported it. So you can very nicely drill down. And conversely, for every gene set, you can drill down to see the genes that supported that. Just some quick validations before I get to the actual results. We're still working on exactly how to validate this. But the top genes, if you know anything about diabetes, you could probably have guessed this, but all of them are relevant to diabetes pretty obviously. The pathways are very obviously relevant, and this is significantly higher than what you'll get if you look at like the top pops and magma gene sets, and exactly kind of validating that for more traits. And kind of exploring the reasons why is something that we'll be doing. The overlap with the curated list is much higher, fourfold as many in this full list of gene sets relative to POPs, and fivefold higher for these very, very clear candidate causal genes. And then two other things we've done is we've looked at whether the top scores are enriched for independent rare coding variant associations. Here I'm showing the top 50 ranked PIGEAN genes, and then the p-value percentiles for rare variant associations. And you can see those are shifted upwards and are significant. And that's higher than what you'll observe for POPs. And if you asked why I chose the top 50, I didn't have to. This is a plot that's kind of showing the enrichments for all possible numbers of top genes, and you can see it's pretty significant for most of these, which again compares favorably to POPs. And again, I'm not trying to, POPs is just kind of the only method out there that we can compare to. We're not trying to, like I said, I think POPs is a very valuable tool, and the fact that it's used so widely indicates that. And we're trying to extend those ideas to this more general genetic support question. One final question, this gets at the question that asks, can you use this to kind of support genes that are not genome-wide significant? Okay, so this is an analysis that addresses that. So this is now taking that 2018 T2D GWAS, and then taking all the lead signals that are not genome-wide significant. Okay, so these are p-values above 5 times 10 to the minus 8th and below 10 to the minus 3. So it's not like everything, but below anything that's reasonably significant. And now we take our 2023 T2D GWAS from the portal, which is much larger. Okay, so it's kind of like we're going back in time and asking, if we ran this in 2018 and predicted things, what would we find in 2023? Obviously, the strongest predictor for whether something is significant in 2023 is the 2018 p-value. So that is very, very clearly significant, right? But even controlling for that, if you add in the PIGEAN score, you do get highly significant predictive ability that the genes with the highest PIGEAN scores are much more likely to increase in significance when you go to the future GWAS. And that's exactly what we're hoping to see, right? We're trying to distinguish those insulin receptor genes at 10 to the minus 6th from other genes that have nothing known about them at 10 to the minus 6th. What's also interesting, although I don't have time to get into this, is that even the genes that are already significant increase in significance more than when they have higher PIGEAN scores. And I'm not exactly sure what that means, but that is something that we'll also be looking into. So all of that, I think, has convinced us that even though we have more validation to do, and we are interested in, we're trying to get this out as a preprint very, very soon, there's enough. We're confident enough in the results that we're comfortable releasing them internally to some early adopters who might be interested in giving us feedback on them. And so there is an internal portal I'm going to show momentarily that we've applied this to the 1,011 traits in the Association to Function Knowledge portal. As one thing is, this is only based on GWAS. It's trivial to incorporate exomes data. We just have not had time to do that yet. So this is only going to calculate support from GWAS and then all the indirect support that we have. And in terms of where the annotations come from, there's actually 3 sources. You can select by default just mouse data and pathway data. You can add in additional gene expression values as well as interaction partners. And then if you want to go for everything, you can add in literature as well. The literature, we're just uncertain on of how much you want to include that in the model, because the literature is enriched for a lot of genes that people think are relevant. And it's certainly, you're able to predict GWAS signals well, but that's in part because the literature is enriched for genes that are in GWAS signals. So that's kind of like the largest one. And, I'd recommend starting with the small set when you're exploring this. And then there's this other parameter I won't get into, but I'd suggest leaving this as medium, which is just a default parameter that's probably not that of interest to most people here today. Okay, so this URL, which we are not going to send out, but I suggest you write down or take a screenshot if you're interested in, is the URL you can play around with this at. It's at our portal, :8000/PIGEAN. And actually any portal on our network you can use this for. So if you just wanted to use this common metabolic disease knowledge portal, you could just do hugeamp.org. If you wanted to use the cardiovascular portal, you could do cvd.hugeamp.org. Any of the URLs with an 8000 after it /PIGEAN should take you to this. And I will now switch to that just to kind of take you through some of the data you can explore. And so you can search, so there's 3 pages. There's a phenotype, a gene, or a gene set. And so I'll start with phenotype, and I will load type 2 diabetes, since that's what I've been talking about. And you have 2 cards that you get. And so, by the way, on the top here, that's those 3 gene sets that I recommended. I'd suggest defaulting that to medium. And then I'd just suggest this is the number, that's small, medium, large. I'd suggest for small. And so, but you can play around with that if you want. There's 2 cards. There's one that gives you an evidence of the top ranked genes, and then there's the top ranked gene sets. And I'll kind of just go through what these columns mean. So this is not, we're not releasing this because it's not documented and it's not obvious all of what these things are. So I'm just going to try to go through this. I'm happy to answer questions, but I think it's more intuitive once you play around with it. You'll see how it works. So you have a list of the top ranked genes, and they're ranked by this combined score, which is the combination of direct and indirect support. The units of these are the log of the huge scores. So if you exponentiate these, you'll get values that are comparable to what are currently on the huge page. And we'll probably add the color coding to this page once we release this officially to the portal. But that's the combined score. And then the GWAS evidence weighted is your direct evidence. That's why it's called GWAS. And then gene set evidence is your indirect. The unweighted score is of interest because this tells you whether this is like kind of just based solely on proximity to a signal. That's like the existing huge score. And the GWAS evidence weighted tells you after you know all the annotations for the genes in that locus, what that gene looks like. And I think I have an example for like insulin. Yeah. So if you'll see, insulin is an example of a gene that the unweighted score is about 2, and then it goes up to 5 once you account for the annotations. And that's because initially this signal here is closest to this gene TH. But once you know that insulin has all these highly relevant diabetes annotations, it thinks that that's much more likely to be the causal gene at that locus. So the GWAS weighted is kind of like the integrative method of kind of what POPs is doing, but it's incorporating both proximity and the annotations in a very probabilistic and statistical way. Okay, so the scatter plot at the top kind of shows all of the genes plotted by their indirect evidence or gene set evidence on the x-axis and their direct evidence on the y. And so, like, for example, you can look at genes down here like IRS2 that have a lot of indirect evidence, but not a lot of direct evidence. Genes on the top like RD55 that don't have any gene set evidence but have a lot of direct evidence. And then the genes kind of in the upper diagonal have a lot of both. And these are like the ones that are going to appear at the top of this table. Okay. And you can do things like sort the table and filter it. So, some cool things you can do is if you want to say, like, I want to get rid of, all of the genes that have any, I'll do weighted, any GWAS evidence at all. Okay. Like these are kind of like your top novel genes, right? So like there's very, very little known about these genes, at least according to the gene. Oh, sorry. Yes, no, sorry. I'm doing the opposite right now. These are genes that do not have a signal, but there's a lot that are known about them. Sorry. So NR1H4 does not have a GWAS signal, and I think I have that locus here. So there's like literally nothing in the region at all. The top p-value is like 10 to the minus 4, but there's a lot of gene sets that are known about it. And you can see that by clicking on this show button here that will show you, okay, it's involved in lipid homeostasis and free fatty acid level and circulating insulin. These are the mouse gene sets. These are the pathways. And so there's a ton known about it, and yet it doesn't have any association nearby. So those are kind of like the kind of what's going on with these genes. What I meant to do was to actually filter on the gene set evidence to be very small. So if we set that to be small, these will be kind of like your genes that don't have any gene set evidence, but have a very strong like evidence of association. So there's kind of like your novel genes that not much is known about. And I did notice KLF14, which for those who know diabetes are quite well known, but for whatever annotations it has, those were not picked out to be important. So now below, then you have the gene sets, and here the scatter plot is now showing that marginal and joint effect sizes that I mentioned. So a lot more gene sets have marginal effects. There's that KEGG maturity onset diabetes of the young that has an effect in isolation, but is better explained by other annotations. And then, the ones that are higher on the y-axis have, are significant after you control for them. And so again, you can expand these and see the genes that went into it. Okay, so then there is a gene page. So you can access that by typing in a gene on the home page or just clicking on a gene from there. And now you have your PheWAS plot that is just like we have on the current gene page, but now it's the combined score, okay? So you have to be a little careful because some of these phenotypes are significant because of indirect, some are significant because of direct, and some are combined. And this scatter plot kind of gives you that at a glance. So you can kind of hover over here and see that insulin is very likely to be involved in HOMA B based on the gene sets it has, but it doesn't have any direct evidence. Whereas it has direct evidence for testosterone, even though there's no gene set or there's minimal gene sets linking it to that. Okay, and so again, this is like a more continuous version of the PheWAS plot we have now, and you have the same table, and you can again see like the gene sets that were responsible for this gene being involved in that phenotype. And then there's a gene set page that if you click on that will give you a PheWAS of that gene set across all of the phenotypes. And again, you'll get this plot of marginal versus joint. So the higher marginal are those that there's some relevance to the trait. The joint is it's significant after controlling for all of them. So those are like the main 3 pages on the portal that you can play around with. And let me see here. I wanted to give some kind of like, tidbits for this, which is that again, so GWAS unweighted is what you should use for the direct. Gene set evidence is the indirect, and combined is both. When you're looking at genes, make sure to like kind of parse out traits into direct and indirect. Again, those are 2 different concepts, and the combined score gives you the best single summary we have. But there's a difference between genes that have direct and indirect support that hopefully should probably be intuitive to most of you. And again, as I mentioned, all the values you can the same are the log of the units on the huge calculator. And if you just want a handy kind of guideline, it looks like 3 is very strong, 2 is suggestive, and then 1 is like nominal. Anything below 1 would be weak. That's for genes. For gene sets, again, make sure to consider whether you want to know the joint effect versus marginal. Sometimes joint is most interesting. That's mainly for predicting, explaining why a gene has high support, whereas the marginal are most useful for a PheWAS. And in terms of interpreting these values, typically something above 0.01 is actually a significant effect. And once you get above like 0.5, this is a very, very relevant gene set to that phenotype. So just a couple things that we've noticed here, and then hopefully I'll have a couple minutes for questions. What's been remarkable is like the animal data that we've looked at, the mouse phenotypes are highly, highly sensible for almost every trait you look at. So like chronotype, morningness, the top one is shortened circadian. It's like almost a direct one-to-one map between the human phenotype and the mouse data. And that's not always obvious if you just ran Magma. This really becomes very clear once you are re-estimating the genes and gene sets using this method. Again, it's kind of showing here, this is what you would get if you just ran Magma. It's not like it's not that it makes no sense. You see KEGG circadian rhythm manual. It's just that it's much more enriched for kind of less clean stuff than you would have here. an interesting thing we're going to look into is like how things change based on what evidence source you put in. So here I'm showing like the top indirect from mouse alone for diabetes versus pathways, and you can see it's not always the same. Here, text versus gene expression. So gene expression, you'll find genes that are like very, very active in islets for the type 2 diabetes. And on the x-axis, you'll get a lot of like drug targets because that's been published on a lot. Usually, you know, when we fit the full model with everything, you often, you know, kind of add support for every gene, but there are some cases where genes that are supported only by mouse data are not supported. That's what's shown here on the x-axis once you do all of the annotations, because there are other annotations that explain those mouse ones better. Some interesting questions that I found interesting is like if you look across all traits in the portal and ask like what genes have the most relevance to the traits in our portal, you kind of get like you can kind of get at a glance like the genes that have high prior and high direct GWAS evidence. So like PPR gamma is the one that's kind of the most, and that's partially because just these very well studied genes, but also because we're enriched for metabolic traits in our portals. But you can kind of use that to zero in on like some genes like PD3A, where like there's very little known about it based on like the lack of indirect support for a lot of these traits, but there's a ton of GWAS hits for it. So I think that's just what's going on with that gene. That's kind of interesting. Whereas on the other extreme, there's a gene like COL1A1 that has extraordinary support from gene sets for like a very, very, very large number of genes, phenotypes, but very rarely do you see a GWAS association observed. And if you look at the nomad constraint for that, that's a very highly constrained gene, and that might be one of the reasons why. And this is why indirect support is interesting to take a look at, because this gene is probably relevant to these traits. You know, a lot of these are the musculoskeletal traits that you would expect, but you're not observing a GWAS hit because there's no, it's a very highly constrained gene. So depending on what you're studying, it may be of interest to see this. And then finally, I think this distinction between direct, independent, and dependent of gene sets is quite interesting. So if you look at coronary artery disease and you look at like the top marginal gene sets, you get all these cholesterol genes. And that's what you get if you do like, you know, any traditional gene set enrichment analysis. But if you look at the top jointly fit ones, you now see less overlap amongst mechanisms. And that's because, you know, a lot of these other cholesterol mechanisms here are redundant with each other. And you can actually see that this factors almost into cholesterol and then something that's more about the structure of the heart and then about vascularization. And for the, I mean, I'm not a CAD researcher, but my understanding is there's great interest in parsing out like lipid mechanisms from vascular mechanisms. And you can immediately do this by kind of looking at the genes that are grouped under this cholesterol gene set compared to the genes that are grouped under this other mechanism. And they're very, very largely non-overlapping. And these genes, you know, could be immediate candidates. If you're browsing the portal, you can kind of just select the gene set that's the mechanism you're interested in and kind of, you know, I'm just showing two here, FOXC2 and PGF, which are not on the effector gene list that we have for coronary artery disease. In the case of FOXC2, that's because it's not actually in a GWAS locus, but it's near genome-wide significance. But then it has this prior information, a lot of it about the heart structure and vascularization. PGF is in a GWAS locus, but for whatever reason is not on the effector gene list either. And these are just kind of examples of how PIGEAN's pulling out genes specific to a mechanism, both within a GWAS locus and outside of it. Anyway, so I have 5 minutes left just to tell you next steps. If you're interested in perusing this or even helping us validate and publish this, we actually need a ton of help in just putting this together into a publication. And we are actually, believe it or not, actually looking for help with that. So please contact us and we'll obviously be documenting this and putting it in the portal more thoroughly. But in the meantime, if you're comfortable working with a site that's not that well documented and playing around with it, this is the URL again, and you can write it down and play around with it. So let me take a question. So there, question from Alan, there's a huge amount of data in RNA-seq of single gene knockouts. Can these be incorporated? Absolutely. Any feature that, any value that you can measure across a subset or all genes, it's trivial to incorporate. You just need a file that lists genes and their values. And then the way the model works is that it's got what I briefly touched on. It has very, very significant sparsity constraints. So it's very, very robust to including hundreds of thousands of annotations. So you can throw in tons and tons of that data, and it will select out which ones matter. When we've incorporated the single cell data from human and mouse and all, like, so the POPs method has compiled like I think 50,000 of these single cell. When we feed that, there are some traits like diabetes, which actually that information doesn't help a lot because it's redundant with all the others. But there are other traits like type 1 diabetes, where it really adds a lot. And obviously you might be interested in prioritizing genes solely based on single cell data as opposed and taking out all the text mining and stuff. And so yeah, and Avi, like, so and that would be definitely interested in talking about that. That would be really cool. Marginal versus joint effect. What is adjusted in this model? Other genes or anything else? So that is all of the gene sets. So you can think about it as I'm trying to predict the gene probability given all like 50,000 gene sets in my mod that I have. The marginal would be I fit each gene set one by one in isolation. That is the marginal. So that tells you whether that gene set's relevant to the trait. The joint effect is once I condition on all other 49,000 or whatever, what's its now residual effect? So you're jointly fitting all the gene sets together. And that's why you see, like such a difference when the marginal effects, you see all these cholesterol gene sets because all of them are significant. It's just that they're very, very highly correlated with each other. So when you jointly fit them, statin inhibition of Wiki pathways cholesterol metabolism kind of explains all of the others, or to a large degree, such that like they fall way down below these other gene sets disorganized myocardium that are more independent. So that's kind of how that's working. And we have some, if you're interested, we have some very early results about going a step further and actually clustering those jointly fit effect sizes to kind of tease out independent mechanisms, which is looking quite interesting. So we're almost at time, and please pop any other questions in the chat, but I just want to make sure to thank the portal team, the lab, obviously everyone who's, Mackenzie and Noel and Maria who run these webinars, but particular thanks to Annie Moriando, who did all of the direct coding for the site, Patrick, who ran all of these, all the results across our portal, and then who also, oversaw a lot of the work that Annie did and helped out too. Okay, I think we have any other questions or not? I don't see any in the chat, and I think we're nearly at time. But thank you all for coming. I guess this is this is a little bit of a departure. This is not something that's yet in the portal, and it's kind of blurring with the research. But we are, it's something that we are hoping to roll out. Hopefully, at the latest would be our October release. This should all be up in the portal, and hopefully you'll see a bioarchive for this method sooner than that. All right, I think that's probably it. Thank you everyone for attending. Please email me for questions if you want to see it one more time. There is the URL and either our help list or me directly if you're interested more in the statistics or the validation of the model. And thank you for all of your time.